Okay, case study 4 on muscle physiology. This particular one deals with a couple of different scenarios. At the beginning, you'll be asked to match up from a word bank some different terms, important terminology when it comes to the micro construction of a muscle. Call it a muscle fiber, it's a muscle cell. So we'll see some terminology associated with the specific muscle fiber cells. We'll look at, or you'll see some of the different steps involved in getting a muscle to contract. So anyway, there's a little section there that says you're responsible for giving a little tour to of the clinic to some high school students. So anyway, there are the definitions there. And then the word bank is up here. You'll use each one of those once. And then the next, we get a patient next. So the first one is Jeff, and discusses, in essence, that Jeff works in a lab for the CDC, and he was exposed to sarin gas, a little bit of liquid splash onto his arms, and then after that, he was dizzy, heart started pounding, shortness of breath, sluggish, etc. He basically falls, his muscles cramp up, and he falls down and hits his head and is unconscious. So he's rushed to the emergency room. And so it asks about the symptoms he's experiencing. And then it asks a question about ACHE, which is acetylcholinesterase. That's the enzyme that kind of breaks down and enables the reuptake of acetylcholine once it's left the neuron, or this what we call the synaptic knob, the presynapse. Once that acetylcholine leaves the presynapse and it attaches to the receptor sites of the postsynapse, the motor end plate, or the muscle cell or muscle fiber, we're going to see a series of functions or dominoes, in essence, start to fall. So when we're ready to relax a muscle, we need to get acetylcholine back to the synapse and ultimately back to the presynapse. So acetylcholinesterase, you have that ASE on the end. This is how it's abbreviated, ACHE, is going to enable acetylcholine to go back to that presynapse and enable or allow a muscle to relax. So yeah, it's going to ask a little bit about that. We get a chart of Jeff's test results. We see the acetylcholinesterase is at 44 and should be at 100. So he is low on acetylcholinesterase. Okay, so it asks about that, which are abnormal. The mechanism of action of sarin or sarin-like chemical. So you can research that. I also, over in your Blackboard, there are notes for that assignment. So let me go down into case study assignments. And if I click on case study for muscle contraction, there's a little notes area. So here's what's going to come up. First of all, this is going to give you the question. So you'll click that and then you'll see where you're going to type in your answers. It has the matching and the word bank, so you can type everything right into this document. And again, remember with these this is a Google Doc, so and it's non-editable, it's read only. So the only thing you're going to be able to do is click File and either make a copy or download as a Word doc and then type into that. So anyhow, that's there. And then if you scroll down a little bit, there are going to be some case study for notes. So if you click on that, Okay, it's going to give you just some notes that'll help you answer those questions. And one of the notes in there talks about sarin gas and a little bit about what that is. So so in essence, you're going to want to use these notes. There it is, sarin gas. And then the next one, the next patient goes into myasthenia gravis. Okay. And there are a couple references with websites. Okay. So you can check those out. Definitely going to want to use those notes to do this particular case study. And then if you scroll down a little further, muscle contraction PDF, that's going to be the actual case study. That's what we're reading right here. All right. So anyway, those notes will help you answer these questions. So, be as descriptive as you can. Now, again, it talks a little bit about the process of a muscle contraction. So again, we get acetylcholine from the presynapse which is the nerve, to the postsynapse, which is the muscle. And then we're going to see calcium released from the sarcoplasmic reticulum. And at that point, we're going to see troponin and tropomyosin connecting with that calcium and enabling the binding sites for myosin to be exposed. So when acetylcholine is released, calcium is released, and actin and myosin are bound together, causing a contraction. So we need to go the reverse way. We need to get calcium back in the sarcoplasmic reticulum and acetylcholine back into the presynapse in order to get a muscle to relax. So in this situation with sarin gas or sarin poison, we see that Jeff doesn't have any acetylcholinesterase, or he does. It's not enough, but it's causing his muscles to stay contracted. So anyway, you can discuss that a little bit. And then the next patient, the last part, is Sandy, discusses a little bit of her symptoms. So you'll want to go through those, type those in. Okay. And we look, everything's pretty normal, it looks like, except for the last one, antibodies for ACH receptors. So she has the opposite issue. She has an autoimmune issue where her antibodies are not allowing acetylcholine to bind to the postsynapse or to the muscle cell. So she's unable to get muscle contractions to occur the way they should. So anyway, you want to go through and answer those. You do not have to do the table. And I mentioned that there's the case study notes and then the questions. And at the very bottom, it says you do not need to do the table. So anyhow, that's it for that case study. And again, if you have questions, please email me and I'll help you lead you to the right direction.